Good afternoon, everyone. Welcome to today's weekly research conference. The Heart Institute is proud to celebrate its 50 years of innovation and excellence. These conferences continue our mission to advance cardiovascular science. We're honored to have Dr. deKemp come and talk to us today for this in-person conference. And I guess it's today the first time that it's only in person. Anyway, last week was the first time? Okay. So going forward, this is only going to be an in-person event, so it won't be online. So the people who are here, you are the audience. However, it's being recorded, so these conferences will be available afterwards for online viewing. There's a link here for people who want CME credits. That's what you see up there. So any questions after Dr. deKemp's finished his presentation, please use the microphone so that it will be recorded for posterity, your question. And now I'll introduce Dr. deKemp. So Dr. deKemp is known probably to many of you here. I see a large contingent of imaging people in the audience, but he's the head imaging physicist in cardiac imaging at the University of Ottawa Heart Institute. He's associate professor of medicine, engineering and physics, medical scientist in the Faculty of Medicine. He also holds an adjunct professor of physics and a member of Faculty of Graduate Studies at Carleton University. In terms of his prior training, he trained a Bachelor of Science in System Design Engineering at University of Waterloo. Then he went on to a master's in medical physics and PhD in electrical and computer engineering at McMaster University. Then he came here. He's been here since 1994, and he helped establish with Dr. Beanlands the National Cardiac PET Center at the Heart Institute. Dr. deKemp is a leading expert in physics of cardiac PET imaging. He's pioneered many applications for Rubidium PET perfusion imaging in Canada. When I was in Edmonton, he helped us set up our PET perfusion program out there. So he's really been instrumental in helping a lot of programs get up and running across the country. It's been in routine use, Rubidium perfusion at the Heart Institute since 1997. So what's that work out to be? Almost 30 years, I guess. He's past director of Ottawa Medical Physics Institute at Carleton University, editorial board member of the Journal of Nuclear Cardiology. We're going to hear about some of Dr. deKemp's interests, but he's interested in medical imaging physics and engineering, including PET, single photon emission computed tomography, so SPECT, X-ray, CT, multimodality fusion imaging. And he's also developed innovative tracer kinetic and statistical parametric methods for imaging blood flow on PET and metabolism. He's done 3D fusion imaging, coronary angiography. And I think we're going to hear mainly today, though, about AI, new physics and AI models for Rubidium PET in cardiovascular disease. So with that, I'm pleased to hear Dr. deKemp. So,
thank you, Dr. Patterson, for that kind introduction. It's always embarrassing to try and summarize a 30-year career in a few minutes, but thank you. I tell people when I work here, I kind of masquerade as a physicist. As you heard, my training is really kind of engineering in PET imaging, but different domains over the years. Just going to get that pointer. Oh, nice. Okay. Title of the talk that I gave a couple weeks ago, Physics and AI Models Applied to Rubidium PET Imaging. I'd like to use this in the past. These are images actually of the first black hole images that were published initially at the Event Horizon Telescope. It's what the PhD is in 2019. And, that kind of looked like I used to put kind of SPECT images up in comparison to this. They're kind of low resolution. But over time, these have been improving. Even just last week, I saw updated images, in fact, using AI models to help improve the quality of these physics and astronomy images. And Likewise, there are improvements happening to PET images in that same kind of 5-year time period. Now, sadly, I'm going to have to pull a bit of a bait and switch, because when I started to put the talk together, I realized that there was actually quite a bit of physics to talk about. So the talk's going to be mainly oriented towards physics, although there'll be maybe a couple of teasers on some of our AI work towards the end. So these are my disclosures. We've got Rubidium PET Technologies licensed to Jubilant, and they've actually become one of our good industry collaboration partners, also together with Siemens Imaging, in particular, since obtaining the new Siemens scanners a few years ago. They're very open, the physics and engineering group, to research and collaborations. So just to summarize, for those who may not be that familiar with PET imaging, the use of imaging, nuclear imaging in particular, in cardiovascular disease. Our use of PET is mainly for diagnosis and management of patients with coronary artery disease. There are a few PET imaging tracers, and I'll talk about Rubidium, obviously, but also one of the new ones in green, flurpiridaz, which is emerging. Other indications: viability for bypass and transplant stratification, inflammatory diseases where FDG, labeled glucose, has been front and center for many years. There's a new tracer developed by Adam Schuhendler at Ottawa U, fluorodeoxyfructose, and we're collaborating with him. Keep an eye out for that. Maybe a game changer in the domain of inflammation and infection. And other tracers produced here in the radiochemistry lab: sodium fluoride for active calcification, potential interest in new tracers. One of them that's particularly promising, FAPI, looking at fibroblast activation. In this talk, I'm gonna focus mainly on some of our progress and some challenges with Rubidium PET imaging. So there are a number of imaging modalities that are used to in the diagnosis of coronary artery disease. We say CAD. At the top of the list, most commonly used in North America, is SPECT imaging. Number of procedures, around 2 million procedures per year. Notice there's a break in the Y scale here. The next group are among them PET imaging, which has been fairly steady, maybe increasing slowly over a number of years, and stress MR also becoming increasingly used, as a nod to Dr. Patterson here. When we compare the PET versus SPECT, though, in terms of stress, say the nuclear imaging modalities alone, PET is still historically a relatively small player, but even in the last 5 years has essentially doubled from 10 to 20% of nuclear procedures, and continues to grow. This is recent data from Moaz Al-Mala from the US Medicare. Similar would be in Canada, although obviously 10 to 20 times lower volumes. So our involvement with Rubidium has been ongoing since I came here. Dr. Beanland, Dr. Ruddy were instrumental in helping to bring this technology to Ottawa. And around 1997, we did our first patient scan, as Dr. Patterson mentioned. How does our use in Canada compare to those numbers in the US? So after 10 years of use, we licensed this technology to a company in Montreal called Draxamash. And it took them about 5 years to kind of take that technology, implement it, start to scale it up, get Health Canada approval. Around that time, we had the Rubidium Army trial. You may remember the isotope crisis of 2009 that helped us to kind of promote the use of Rubidium, including Edmonton, that was one of the participating centers. But things really began to take off around 2016-17 with FDA approval, and the company since obtained approval in the European market as well. And there are, I think about 100 sites now being supplied with this generator. One other company in the US, but given that the overall PET volumes are increasing slowly, clearly the Ottawa technology is rapidly gaining market share in this domain. Also around that time, our professional society, American Society of Nuclear Cardiology, or ASNC as we call it, realized that cardiac PET was really being underutilized at that time, and put out this information statement, or this position statement, emphasizing that PET really should be recognized as a first line and preferred test for patients who would otherwise be referred for pharmacologic stress testing and imaging. The complement of that is really had not been applicable for those who were able otherwise to complete a treadmill exercise study. And treadmill exercise, for those patients who can run on a treadmill, has important diagnostic value in terms of ECG changes. Time that they're able to run on the treadmill is very useful in terms of severity of their symptoms. So here's what a kind of PET system looks like for a rubidium stress test, thanks to one of our technologists who took this photo for me last week. This is in fact the rubidium generator in a small case. It's positioned right next to behind the scanner with ECG and blood pressure monitoring equipment. And rubidium has a very short half-life, 1.25 minutes, basically requiring us to have the patient on the scanner bed while the tracer is injected. And for that reason, imaging at stress, we use this pharmacologic agent to simulate exercise, because there's no time for the patient to run on a treadmill, inject. By the time we got them into the scanner, the tracer would be all gone. So here's what a typical protocol looks like. Patient comes, they're positioned in the scanner. We CT scan for attenuation correction and anatomical localization. We give this vasodilator, which is usually adenosine based. Adenosine is a smooth muscle vasodilator, so it relaxes the coronary arteries directly, allowing blood to flood into the heart muscle. And then we inject again and take pictures at stress. And because of the short half-life, this whole test can be completed in some centers in as little as 20 to 30 minutes. The PET scanners today are actually hybrid scanners, combined PET with CT. And you see here images on the top row, acquired post-stress, and on the second row, the resting images. Bottom row is fusion. CT images are shown in grayscale, and in color here, with these kind of cool colors, is actually where the rubidium ends up in the myocardium. And the idea is basically in a normal heart, you would have basically homogeneous intensity of tracer, and this is a relatively normal scan with uniform, horseshoes and circles overlaid with the CT. Conversely, this would be an abnormal scan. You see a patient. Normally, we rotate these images. The heart sits at an oblique angle in the chest, so we rotate them into a standard view where we can cut in short axis and long axis cuts, which are more closely aligned with how the coronary arteries supply blood to those tissues. And in this case, we see here on the bottom row our rest, on the second row our stress, and we can see at the bottom here, maybe with the yellow arrow, kind of a little chunk or a little bite taken out of what we would like to see as a nice homogeneous donut. Or also on the bottom of the long axis slice, you can see that intensity on the inferior wall is relatively reduced, and this would suggest that there's disease in the artery supplying the right coronary artery in this case. So what about treadmill exercise? How does that differ from what we do typically in PET? So ECGs would be placed, the leads would be placed to monitor 12-lead ECG. Patient would run on the treadmill. When they're getting close, they're like, okay, I'm good, I can't go anymore. We inject a tracer to kind of capture how much blood was being delivered to the heart muscle at that time. And then after some time for recovery, we would image the patient. So we get combined information about stress ECG, time on the treadmill, plus if a tracer is injected, distribution of perfusion during the time of exercise. And up till now, this has been the domain of SPECT imaging. And the main reason for that is because SPECT uses isotopes of technetium-99m most commonly, and it has a 6-hour half-life. So there's time for the tracer to be injected. You've got a couple hours, an hour, half an hour to wait. The patient can go. The tracer is trapped in the muscle at that point, basically as a snapshot of perfusion, and we can take the pictures sometime later. So it makes it feasible really to do post-exercise imaging. The PET isotopes, if we wanted to try and do this with PET imaging, what are the isotopes that we would have available? Well, the most common used today is rubidium, but as I mentioned, it has a very short half-life. It comes from a generator, which means we can, elute with physiologic saline and put this device next to the patient bed. So it's convenient, but really too short to use for exercise. Among the other isotopes, really the only one that would be practical is fluorine. We have fluorine-18, fortunately, with a 110-minute half-life, about 2 hours. So on the same range or on the same time scale as technetium. Cyclotron is a much bigger infrastructure than a rubidium generator, but regional cyclotrons exist, particularly through the US. They make labeled fluorodeoxyglucose and distribute F18 labeled tracers around the country. And similar could be done for a PET scanner. The other property to take note of is this thing that we call positron range, and I'll come back to that when I'm giving you a little explanation of some of the physics. But you'll note that rubidium has a pretty high positron range, and rubidium is much smaller, which is a characteristic that's better to get higher resolution images. So could we potentially think about a PET imaging tracer for perfusion? Recently, or even not so recently, the past 15 to 20 years actually, there's been a class of tracers being developed based on mitochondrial complex 1 inhibitors, rotenone derivatives. And flurpiridaz is one that has made it the furthest in terms of clinical testing. Compared with rubidium, which enters in via a sodium-potassium pump, or ammonia, which is freely diffusible, but then is metabolically trapped in glutamine, flurpiridaz is a very strong inhibitor of MC1 and has almost irreversible binding within the mitochondria. There's a lot of hype currently in the nuclear cardiology field in terms of what's going to be the potential impact. This hype has been going on since 2017, but finally we have a successful phase 3 trial that was published in 2023. This was a large study, 700 patients from 48 different sites in the United States, Canada, and Europe. Dr. Beanlands was on the steering committee and led recruitment of patients in Ottawa. We participated in this trial. And, the conclusion, flurpiridaz has utility as a tracer for CAD detection. SPECT, in this case, was used as the tracer, as the standard for comparison. It stresses the most commonly used nuclear medicine technique in the US. And this trial was aimed at FDA approval in the US market. And what we see here when we look at these tests, these are what we call receiver operator characteristic curves. They basically show the utility of the test. A useless test would fall somewhere along this diagonal line of chance, 50-50 chance of getting the correct result or not. A perfect test tends to be up, or a better test tends to be up in the upper left corner. And as we can see, PET overall and in certain clinical subpopulations tended to be better, or was at least equivalent or sometimes better than SPECT in virtually all cases. This then led to FDA approval shortly after, September 2024, and GE launched this tracer commercially at the ACC last year in March. Again, lots of hype in our area. The first new nuclear perfusion tracer approved in 30 years, SPECT or PET. I don't know what it would cost to get, but it's, on the order of $50 to $100 million to get a new tracer like this into market, on the order of new pharmaceuticals. So we have now some new first case images coming out from users, independent users. The exercise perfusion protocol looks something like this. We give about 3 millicuries, a relatively low dose at rest. Wait at least 60 minutes, because remember, this has a 2-hour half-life. So we want to give some time for that rest residual to decay so that we can see any stress abnormalities or any abnormalities on the stress pictures. Patients run on a treadmill, some time for recovery, so the breathing will slow down, and then take the pictures. And these are the kind of spatial resolution now that we can obtain with PET. Compared with MR or CT, they are still, low resolution images, but they are, probably among the best perfusion images that we'll see now. This was a patient with a prior disease in the right coronary artery. They've been revascularized. There's maybe still a little bite out of there in the inferior wall, and potentially some new or very mild disease in the anterior wall and septum that the authors claim to have seen. Unfortunately, there was not cath correlation in this case to be able to verify. But, exciting use of this new tracer in the clinical field. More interesting, again, from Amala's group, Texas at Methodist Hospital, one of the first comparisons of rubidium with flurpiridaz. So these are very typical of the kind of resolution and contrast that we would get with rubidium. Again, stress images on the top, rest images on the bottom. And if we move same patient scanned with flurpiridaz, you can see, better definition of the walls. If I go back to rubidium, walls look thinner, better recovery. The blood is very black, so the contrast is better between blood and tissue. And when we look at the kind of net advantages or some of the differences in the head-to-head comparison, even just qualitatively looking at this rubidium picture versus flurpiridaz, a little bit better definition of the walls and blacker background. So we talk about resolution and contrast. And those are the things that are motivating some of the physics investigations. So I've spent 20 minutes going through some clinical rationale and positioning where some of my interest has been in terms of helping to hopefully improve the quality of rubidium imaging to, let's say, remain competitive with flurpiridaz. So a little bit of physics. The way that PET imaging works, we have some isotopes which are have a relative deficiency of neutrons compared to protons in the nucleus. And what happens is this nucleus is unstable, and a proton decays into a neutron, and the positive charge comes out in terms of a positron, which is a positively charged electron. It's the antimatter, the equivalent of an electron. And what happens is this positron will actually comes out with some kinetic energy, and it needs to travel a certain distance, kind of losing energy before it can combine with an electron and produce these two 511 keV photons with the famous, Einstein's famous equation, E equals mc squared, right? And this positron range, even though all PET isotopes produce annihilation photons at 511, they have different positron ranges depending on the energy at which that positron comes out of the nucleus. And in fact, rubidium is the most energetic, and so it has the longest positron range among the PET tracers. F18 is the smallest. So remember I said 0.6 millimeters, F18 versus about 6 millimeters, 6 to 7 millimeters for rubidium. And that's really imposes a fundamental limit, or in the past has imposed a fundamental limit on the spatial resolution that we could achieve with PET. These are the energy spectra. F18 is kind of a small wedge here, right near zero. And this blue line is rubidium, which you can see has the highest energy and hence the longest range. So when we first obtained this new Siemens system about 5 years ago now, it had a very good spatial resolution, around 3 millimeters fundamental. And so I posed the question to Siemens. I wonder, is it worth looking at doing some research to try and improve or characterize what is the effect really of positron range on our images? And this is a scan done with a fake heart with asymmetric wall thickness, 20 millimeters on one side and 3 millimeters on the other side. And the relative intensity decreases because the scanner is not able to recover the true activity in that case. And so you can see the kind of pink here, we're underestimating the true activity relative to, on the fat wall, we can measure the true activity. And if we do this with no smoothing, try different amounts of smoothing filters on top of the F18, what we find out is if we add 7 millimeter smoothing to F18, we get about the same intensity here on the thin wall as rubidium. So again, some experimental empirical evidence confirming about a 7 millimeter effect. This encouraged us to kind of dig into the literature and look at, how can we characterize the measurement or the impact of rubidium positron range on our measured PET data. So what happens, this is images showing kind of a cloud. If the nucleus is in the middle, this is a simulation of where the positrons would have annihilated, as they traveled away from the nucleus before producing the annihilation photons. This was intended to be a 3D picture. If we project that down to a 2D, kind of XY, and then into a single projection X, which is how the scanner works, we kind of measure projections looking at one view and one angle. The distribution ends up being kind of a bi-exponential, kind of very pointed and very wide tails. The positron range in water now, if we look at a cross-section through that image again, we got kind of radial distance, F18, very narrow, right? About a 0.6 millimeter effect. And rubidium, according to this simulation and this analytic work, around a 6 millimeter effect. So if we have knowledge of this, and we have some knowledge of the scanner's resolution, which is a 3 millimeter, and this thing is usually what we call a Gaussian distribution, normal distributions, those are everywhere in science. If we combine the scanner's intrinsic resolution, we say that that could be measured with F18, neglecting positron range. We combine it with this kind of pointy thing that reflects the rubidium positron range. We still get something that looks actually kind of Gaussian. And in fact, if we fit the Gaussian curve to that, we get a reasonable fit with about a 6.5, 7 millimeter effect. We underestimate the peak a little bit, and we may not overestimate the amount that's spilling out into the tails. So why is a Gaussian response interesting or relevant? It's relevant for some of the physics that we use to describe the way that these cylindrical detectors work with PET. So when some of our activity is off center, you can see it's, these detectors are long. They're actually long and skinny. And sometimes you can have an event or a photon might not be absorbed here, might scatter and then be absorbed here. So there's kind of what we call a depth of interaction effect. There's some blurring in the response. So when we put a point source in the middle of our scanner, you can picture that these two detectors see a very small profile there, right? And we get an image or a profile of a nice circle. Here though, when we put it off center, you can see that now this point source, it's seeing, a lot of this detector on angle. And this basically ends up producing ellipses instead of circles as you move away from the edge of the field of view. This effect has been compensated. There's a pretty deep amount of physics research going back 20 years to characterize this and actually include it into the reconstruction process. Siemens in particular built a robot to actually scan point sources through the field of view and measure those profiles very carefully. So those profiles have been characterized very precisely for a small point source, a small amount of activity for an isotope with a small positron range. And what they found, the way that this is modeled, in fact, at the center of the field of view, we got nice kind of sharp symmetric Gaussians. And as we move away, we start to get these kind of shifted or skewed Gaussian distributions. And the way that these are modeled are simply with split Gaussians, like one Gaussian for the left half, and one Gaussian for the right half. And the inside ones are going to tend to be wider than the outside ones. And in fact, these were measured, and those width of those Gaussian functions, you can see at the center of the field of view, close to 3 millimeters. That's the best resolution you'll get, close to the center. As you move out, 20, 30 centimeters away from the field of view now, if you didn't do this, you'd have resolution of like 10 millimeters, right? And so this is the way that what we call the point spread function modeling response is included. And by doing that, this essentially turns those ellipses near the edge of the field of view, turns it back into circles, right? So this is really well-established physics. So we have now estimated rubidium Gaussians. We simply combine those, convolve them with the very precisely measured F18 data that we have, and predict, what would be the corresponding effect for rubidium. And again, at the center, we got about a 7 millimeter effect, and those combined effects get worse as you move out towards the edge of the field of view. And the nice thing is that the way this is parameterized is that it can be implemented simply by updating a few coefficients in a scanner configuration file. So we did a few patients. This was a group of 10 patients. We compared a kind of standard reconstruction, time of flight plus iterative ordered subset, right? This is the standard protocol. And then we added the PRC, the positron range recovery, onto that. And, the transformation's not profound, but if you look at the vertical long axis sections, I think you can see that these edges are a little sharper than they are here, and the blood background is a little lower, right? So there's a definite visible improvement. And when we try and quantify that, we got about 30 to 40% improvement in quality, what we would call in engineering terms, signal to noise ratio or contrast to noise ratio. Yep. Computer has gone to sleep. Oh, great. Back to the start. Escape. Slide 35.
Just the laser pointer. And when you try to change the slides using the mouse, you have to create the laser pointer. Otherwise, somehow it may froze the slides. In this case, you have to close the slides and remove it again.
It's dead though. Mouse is dead. Yeah, good idea. Coffee break. Yeah.
Or you change the slides using the arrow instead of the mouse.
I was using the arrow.
Yeah.
Yeah, 35. I can find it.
Okay.
Do I dare? Okay, I'm not going to use the laser pointer. So there's an improvement of 30 to 40%. In terms of, what kind of clinical relevance might this have? What does it mean to have a 30 or 40% improvement in image quality? Well, if we look at nuclear medicine imaging, iterative reconstruction methods, our raw data follows a Poisson distribution. The properties of a Poisson distribution, an estimate of the mean equals the variance. And so from that, we would predict that, for example, if we had double the sensitivity, if we had twice as many counts, our image quality would improve by the square root of 2, or about 1.4. That's about a 40% increase, right? So this increase that we saw, we thought would be related to a twofold change in either scanner sensitivity or injected activity, let's say. This was exciting, interesting to Siemens. We've since filed a patent application, which is under prosecution currently. So what have we achieved with this? This is an example of a PET viability, combined viability study. One scan's been done with rubidium and one with F18, although it's not our flurpiridaz tracer, it is F18 labeled glucose. So we have a question, how many, I'll give you 3 options, we'll make it interactive. You'll either prefer the top or the bottom, or you won't know which one is rubidium, okay? So remember, rubidium is generally lower resolution, right? So how many people think rubidium is on the top? Not many. How many think rubidium is on the bottom? How many think that they'd be equivalent or they wouldn't be sure? A lot of people like rubidium for the bottom. Okay. Well, it turns out rubidium is actually the top image. I thought there'd be more undecideds, but in any case, we're trending in the right direction. Very good resolution. We can see papillary muscles here. We can see some detail even on the right ventricle. Okay, thanks. Some details on the right ventricle with the moderator bands here. This is, I've never seen this before with rubidium. So extending, kind of thinking more about the clinical impact, we set this hypothesis then, if the quality is kind of equivalent to a twofold increase or a twofold change in activity, could we flip this on its head and use this positron range recovery to potentially reduce our clinical doses and maintain, by adding positron range correction, can we maintain our image quality with half the dose, right? So we looked back through some of our quality improvement data and found a cohort of patients who'd had either standard full dose with standard reconstruction or half dose imaging, in fact, with and without the range correction. So here's an example patient who's acquired with a half dose. Again, the changes are most evident when we look at the vertical long axis images. So I'm going to move through a series just to give you an idea of quality improvement. So when we add the PRC on it, you can see we kind of sharpen up the edges a little bit, but amazingly, we also reduce the background. And this is kind of a holy grail in image processing. Normally, when you try and enhance edges, you also enhance noise in the background. This has been able to enhance resolution, but also regularize and control the noise in the blood pool at the same time. These are then the full dose RES images without PRC. So if I go back, we're hoping that half dose with PRC would be similar to full dose without. So I think these are fairly similar. Maybe a small preference, I would have said, for this image that would correspond with a little bit of net benefit. If we do use PRC on the full dose images, now you see kind of the full benefit potentially making it look more like an F18 tracer. So the results in this cohort, small cohort of 10 patients, again, hoping that, rest standard would be similar to half dose with PRC, saw, blood contrast still improving, and our contrast to noise. We would hope that this might have been even 0, but saw still even a little bit of a net benefit if these stats were correct. So a net improvement that was encouraging. However, changes in clinical practice are not based on engineering measurements of signal to noise, and additional validations will certainly be required, including visual assessments, quantification, and ultimately evaluation and validation for coronary disease detection. So we tackled the first step, kind of moving away from engineering metrics to clinical perception, and Dr. Omajenk and Kadoya, our senior fellows, were kind enough to review 45 patients, comparing the standard with the half dose with and without PRC. And we asked them to give their visual interpretation of these images with an ordinal image quality scale, and as I asked you to do, to choose one image or the other, or to tell me whether they were equivalent. So we did this based on the ECG gated images, because the gated images are usually noisier. You can see more of the background speckle in the gated images than you can see in the ungated statics. And we made a kind of ordinal scale with some semi-quantitative indices, ranking them from excellent to poor. So the first outcome of this was a little bit surprising. These are arrest standard image quality. So the average score 4, which was very good on average, which we would hope if we're using this for clinical management. A little bit surprised to see that the half dose with PRC actually had a lower perception of quality, despite the engineering metrics, which suggested they might be a little higher. So we added another cohort. We said, okay, maybe we need to include the same 6mm filter as included on the rest standard. We think that that's still fair. And indeed, we got now very comparable quality scores, p-value of 0.8, whereas there was a highly significant difference previously. When we asked the readers, they both did it, to choose one. So before the smoothing, the half dose was PRC. There was a preference towards the standard images. They were preferentially chosen as being higher quality. But with the added smoothing, then most of them were deemed to be equivalent. Maybe, potentially a small preference towards the PRC by one reader, but certainly not a statistically significant outcome. Uniformity. So we want these images also to be quantitatively accurate. Past studies trying to do positron range recovery, for reasons that are not completely understood, have created kind of hotspots in the anterolateral wall. There's some thought this might be the papillary muscle, but wouldn't make sense to have the anterior pap muscle and not the posterior pap muscle. So we checked to see whether there was any septal gradient or anterior-inferior differences, and found, within the cohort, when we compare quantitatively, the positron range corrected minus the standard perfusion values, there's very little difference. This is two one-sided tests of differences. The differences are generally within 2%, which we set arbitrarily as a level that would thought not to be clinically relevant. Next step will be to find a cohort of patients where we have some either anatomic or physiologic standard. In some work in a different cohort that we did with some colleagues in Vienna and in Copenhagen in Denmark, they found that indeed the positron range correction, they also found improved image quality and was able to maintain the ROC performance for detection of disease. This would be detection for disease of, let's say, more than, actually this was a physiologic standard, so FFR positive studies, hemodynamically significant coronary stenosis. I think in our case, we'll have to find a cohort like this. Would obviously hope potentially to find improved performance if we have better resolution, perhaps, to find smaller or less severe defects. And what's the effect potentially then on our clinical protocol? We did some work a few years ago, and Ahita Tavusi published this paper. In our center, we give rubidium dose that's proportional to, or had for many years given dose proportional to weight. So, higher patient gets more activity. But what we found was when you look at the image quality, that proportional dosing is still ending up with lower quality in the bigger patients. And we found that you'd actually have to give the dose as a square of the patient's weight in order to get uniform or really consistent image quality across all patients. And this is what we've been using since obtaining the new scanners. When we look at the impact of dose reduction, here is how the current in orange is our current dosing protocol. We can see that we're able to increase the dose and implement this exponential dosing only up to patients that are 150 kilograms. That's not bad. That covers probably 95% of our patients. In some places in different countries or some states in the southern United States, for example, you might be more in the higher end domain. And the generator itself is only able to deliver a maximum in the US of 2,200 megabecquerels. In Canada, we have labeling that allows us to go up to 3,000. But essentially, we're constrained by the amount of activity in the generator that we can give. If we were able to cut the dose in half by using this PRC reconstruction, we'd be able to implement this exponential dosing regime all the way up to patients, let's say in the US, over up to about 220 kilograms, which is in fact happens to correspond to the bed limit that the scanner will take. So this is the clinical rationale. Comparing the radiation doses, this is a table that's under review as part of the new PET perfusion imaging guidelines. Current typical radiation dose for proportional dosing is about 1 millisievert, 2 millisieverts, sorry. This would potentially be cut in half down to 1 millisievert. And when we're telling patients or we're recruiting volunteers for research, we often use the natural background activity as a comparator. You're exposed to about 2 to 3 millisieverts a year just by living on Earth from things you eat and from cosmic radiation. And so 1 millisievert would be perceived to be extremely low, again, making it among the lowest or really the lowest nuclear imaging test among those that are commonly used. Water has a little bit lower, but it's only used in very limited ways in Europe. So I have a couple of minutes left. I'll just give you, I promised some teasers on AI. So we also have kind of jumped on the rubidium flurpiridaz bandwagon and submitted this work together with Eric Moulton at Draximage and Mathieu Galarneau in Montreal, who we collaborate with quite a lot. Again, you can see a patient here imaged with rubidium. This was one of the trial patients who was enrolled in the flurpiridaz trial. Again, better image quality, thinner walls, blacker blood. We have been interested in quantifying images of myocardial blood flow. So instead of looking at images, min to max, we look at them on a quantitative scale of mils per minute per gram. And indeed, it seems like we can recover contrast that's very similar to what you would get with flurpiridaz and have the added benefit of quantification in terms of these absolute changes in flow from rest to stress. This is a patient, again, the severe irreversible defect on the inferior wall corresponds with an occluded distal right coronary artery. And then we hope to, we are extending this. These reconstructions take a long time. There are hundreds of thousands of voxels or imaging units on a typical three-dimensional matrix. So those pictures can take a long time, and this is where AI has been helpful. The AI can learn what the patterns are in translating blood to tissue. And once the model is trained on inference, these parametric maps can be produced very quickly. And the nice thing is that in addition to images of blood flow in quantitative units, the model includes a fraction of blood. And so for free, we are now getting gated blood pool images, which may complement perfusion, in some cases just for LV sampling. But in other cases, for example, you can see now the right ventricle dynamics nicely, where on PET alone, the right ventricle, because it's quite thin, is really not very visible. So we are now have performed some initial validation studies looking at RV and LV volumes measured using this technique and validating against MR, for example. So this will be kind of the focus of our work going forward. So stay tuned, hopefully, for some clinical validations of that. So with that, I'll thank you for your attention. Of course, none of this is actually, or very little is actually, my work. You know, we have the pleasure as investigators to come up here and present the work of many of our dedicated and hardworking trainees. So I want to thank them, in particular Anahita Tabusi, who did the exponential dosing work, and Owner and Yoshido helped with the PRC clinical evaluations. Thank you.
Thanks, Rob. This is on. I think we have about 7 or 8 minutes for questions. So if anyone has questions, please, as a reminder, come up to the microphone. I can feel someone's presence behind me. A couple quick questions from me. The I'm going to mispronounce it. The flurpiridaz.
Flurpiridaz. Yeah.
Has that received Health Canada approval to be used clinically?
It's, I don't think, been submitted for approval in Canada yet.
But it's approved in the US.
Approved in the US.
Okay.
Yeah. So we're, with Dr. Beanland's help, we've had some discussions with GE on, you know, trying to help gauge their interest for the Canadian market and potentially become a producer.
And so for me, one of the, I mean, the quality of the images looks quite good, but I think it certainly has a, you know, in terms of safety, there's much higher radiation dose with flurpiridaz compared to rubidium, especially with your dose reduced rubidium. So I would, as a referrer or user, I would have some potential concerns depending on the patient group. Like if it was younger patients, female, I would be more concerned about sending them for flurpiridaz as opposed to rubidium. Those are my thoughts. It's more of a comment. My question is for the for some of the work that you're doing and others are doing. You mentioned throughout the talk about potential for artifacts. So what work is being done to help reduce that? Do you think that'll be with your half dosing? Are we less likely to see artifacts with, or is it more with the AI that we'd be seeing less artifacts like attenuation artifacts or different things?
Okay. What
work's being done to help reduce artifacts?
Okay. So if I could comment on the dosimetry observation, we of course hear this in the nuclear medicine domain often from non-nuclear advocates or preferences. I should not advocates, but of course the radiation dosimetry in children is a potential concern, especially for kids that are going to be getting serial tests over their whole lifetime. But we do always say that the radiation risk is actually hypothetical, right? It's assumed that there would be some potential risk at low doses. And for a patient who needs a medical test to be done to help direct their therapy, right, the risk of not treating the disease is probably orders of magnitude higher, right? It's a deterministic effect compared to a potential stochastic effect of low radiation. So I personally don't support the position of worrying so much about radiation dosimetry in patients who are at risk for disease. In terms of artifacts and machine learning and AI, for the PRC, myself, I always prefer physics-based approaches because machine learning techniques, they can be very good at learning general trends and things that are common across the patient population. But in those patients, if I had to go or if I had to send a loved one for a test and they have something kind of unusual on the outskirts, I really wouldn't want to rely on an AI to produce a correct interpretation in that case. That being said, there are some typical patterns that can, I think, be well learned by some machine learning techniques. And among them, for example, is attenuation correction. Those methods are emerging now such that you can actually estimate the correction directly from the PET data. So for attenuation correction, soon we actually won't need a CT scan, although we do want it still for calcium scoring. So we'll have it anyways. So we'll probably have the physics, but maybe with the benefit to modify that measured scan, maybe with cardiac motion or respiratory motion to get very good alignment. So there is benefit there, but always need to be training on real patient data, not simulated data, and performing, substantial external validations. Dr. Beelans, you have 3 questions.
I do. I have one comment and 2 questions. The first is congratulations on outstanding career, 30 years plus at the Heart Institute, and lots of amazing achievements. I'll tell people that some years ago, we had an external review of research at the Institute, and our cardiac PET program is rated as one of the top 5 in the world. And that was principally because Rob deKemp was the top physicist in the world. I mean, he is amazing. He's the person that even centers in the US come to for problems. I don't think people realize that, and Rob's pretty modest about that. But he is one of the big reasons why we've been able to have such straight and strong leadership in cardiac PET and imaging in general. So thanks for that.
Thank you, Dr.
My question, 2 questions. The first is a specific question on the PRC, whether you've been able to show it can improve the accuracy of flow quantification. And then my other question is really, what would be your advice? You've patented around rubidium. You're still submitting patents. What is your advice to young scientists with an entrepreneurial spirit, how they should go about enabling their discoveries and inventions to impact humanity?
It's a big question. The first question is easier to answer. There are really limited standards for accuracy of flow quantification. So I think the quality of flow quantification will improve. So we'll combine the PRC with the AI methods to do parametric imaging. So we'll have the benefits, hopefully, of improved resolution and then the better contrast as well. Flurpiridaz, I think, is going to emerge as the new blood flow quantification standard. And the data that's coming out now, as much as I'm proud of a paper showing that rubidium can quantify flow, probably underestimates the true flow at stress and overestimates a little bit at rest. So as time goes on, I think that not necessarily the PRC, but flurpiridaz will actually be helping us, hopefully, to consolidate the PET methods to get consistent flow values across tracers. For people that have an entrepreneurial spirit, there are different supervisors that have more or less experience and interest in that. We kind of, as you know, stumbled onto it and took a risk to try and put some technology out there that was picked up. So definitely keep an eye out for it. The granting agencies are looking more and more for potential commercial participation as a way to help bridge basic research and get it out into our clinical market so that it can help our patients. It's not easy and it takes a long time. You saw that we started that in 2007 and 10, 15 years later, we're starting to see the, the big uptick. So it's a long road. I wouldn't be preoccupied by trying to submit patents necessarily, but if you think you have something that's really unique and you've got potentially long-term vision, then it's definitely worth submitting an invention disclosure. Try and get the what's our office called, tech transfer office, to look at it, find some experts who can give you a good read. Even despite our perceived success, don't consider myself necessarily an expert in this domain. Keep your eyes open. I think Dr. Ruddy taught me the word about serendipity, right? So keep your eyes open for things that look like they might potentially have a big impact. If you think they do, then go for it. But I wouldn't be preoccupied about trying to make a commercial venture out of everything.
We're out of time, but there is one other quick question for you.
Wenbin?
Hello, Rob. Thank you. Fantastic talk. So the half-life of Rubidium 82 is only 1 minute. I was wondering when the tracer reaches the coronary veins, have they already decayed mostly, or or they still contribute to the signals that you capture?
Yeah, so I think the circulation time, systemic circulation time, is probably 30 seconds to a minute. So, most of the tracer is extracted during the first pass through, the heart and the rest of the systemic circulation. So there's not much, but there would be a fraction, like a 5 to 10% could still potentially show up in the venous kind of circulation on the second pass. Is that a particular interest or concern?
I think like even on the first pass, would you lose some of the tracer even because you're injecting in the venous, systemic venous side, and then it has to get to the myocardium, right? So still has to pass through. There's some pulmonary transit there, and then
Well, it's got to go through the lungs for sure.
Yeah. So that's going to take 30 to 60 seconds.
20 to 30 seconds.
So what percent of the radio tracer would you estimate make it to the myocardium?
By the time it gets to the heart?
Yeah.
Yeah. So we actually also kind of advocated for a saline push to push it like through the venous return to get it at least to the right side of the heart. If you think of on average 20-second delay, that's 25 seconds, a fifth of the half-life. It's like only a 10 or 20% loss. So 80% of the tracer is making it, I think, to the heart in the first pass.
You use a power injector in CMR.
A low power injection, a peristaltic pump. So we don't put it in quite as fast as CT or MR contrast. But
okay. Thank you very much.
Thank you. Thanks, everybody.